This begins Module 8, Session 2, Carbohydrate Metabolism. This highlights important points in chapters 22, 27, and 28 in your textbook. So in general, we'll look at carbohydrate metabolism starting from carbohydrate digestion. Carbohydrates are approximately 50 to 60% of the average American diet, so they're a large component. Then we'll look at two alternative fates for glucose. First off, glycolysis. Glycolysis is the initial oxidation of glucose, and it takes place in the cytosol of the cell. The second pathway we'll talk about as an alternative for glucose is glycogen production, or synthesis, and we'll focus on the synthesis in the liver and the skeletal muscle. We'll also briefly talk about glycogen degradation. So digestion of starch, lactose, and sucrose actually begins in the mouth with salivary amylase. That digestive enzyme, in conjunction with mastication or chewing, begins to break down these polysaccharides into alpha-dextrins. Within the stomach, the salivary amylase is primarily inactivated by the change in pH. So when the stomach contents move into the small intestine, alpha amylase is added from the pancreas that continues this digestive process. In the small intestine, you have these polysaccharides being digested further into oligosaccharides consisting of trisaccharides and disaccharides. Finally, the disaccharides are digested by a family of enzymes referred to as disaccaridases that are associated with the intestinal epithelium and will digest the disaccharides into the monosaccharide form. Once as a monosaccharide, you can see that glucose or fructose or galactose can be transported into the luminal epithelium, where it can then move out into the periphery. So again, salivary and pancreatic amylase will digest large starches. These enzymes hydrolyze these internal alpha-1,4 bonds to generate disaccharides, maltose, isomaltose, and dextrins and some trisaccharides. The digestion then continues in the small intestine by a family of enzymes referred to as the disaccaridases. And these disaccaridases are specific to the disaccharide sugars. So you can see the schematic here. This enzyme has active sites for digestion of sucrose and digestion of isomaltose. It has a connecting stalk as well as a transmembrane segment anchoring it in the membrane of the small intestine. These polypeptides form two globular domains with specific binding sites for each of those disaccharides. So the primary disaccharides are lactose, maltose, and trehalose. And again, there are specific globular membrane-bound disaccaridases that cleave each of these different disaccharides. Lactase, for example, cleaves this specific beta-1,4 linkage. Maltase specifically cleaves the alpha-1,4 bonds. Isomaltase cleaves the alpha-1,6 bonding, whereas trehalase splits the bond between the alpha-1,1 bonding of two glucose molecules. So once the sugars are in their monomeric form, they're transported from the lumen to the capillaries via secondary active transport. And we reviewed this in Module 2, where sodium ATPase transporters in the intestinal epithelium generate a sodium gradient such that sodium from the lumen is transported with glucose down its concentration gradient into the enterocytes. Once inside, glucose is passively transported to the capillaries for distribution to the rest of the body. Fructose has its own transporter that functions independently of the sodium ATPase transporter. There are also facilitated transporters for glucose, shown here, that function independently of sodium. These facilitated transporters are referred to as the GLUT family of proteins. The GLUT family of proteins is summarized in table 27.5 in your text, and for this specific intestinal epithelium, the isoform is GLUT5. We'll look at the function of the other GLUT transporters in tissues as they are relevant. So once glucose is free in circulation, it can be taken up by the cell and oxidized through glycolysis. The first part of glycolysis, where glucose is converted to pyruvate, happens within the cytosol of the cell. The conversion of pyruvate into reducing equivalents, NADH, that are oxidized by the electron transport chain happens within the mitochondria of the cell. For this discussion, we'll look at the process that takes glucose and oxidizes it to pyruvate. For every one glucose molecule, you generate 2 pyruvate molecules. So glucose is oxidized through glycolysis in 2 phases. There's a preparative phase, which requires 2 ATP, and there's phase 2, which is energy generating. This portion of glycolysis can take place in the absence of oxygen. Anaerobic glycolysis, as it's termed, will convert pyruvate to lactate, and we'll look at that in a few slides. So the initial step and the first regulated step in glycolysis is the phosphorylation of glucose. This can be done by either hexokinase or glucokinase. Glucokinase specifically is active only in the liver, whereas hexokinase is specifically active in all other tissues. The affinity of these 2 enzymes for glucose differs, and that's a function of their roles in each of the tissues. We've talked about this in a previous module. The phosphorylation of glucose is irreversible and traps it within the cell. From there, glucose has several different fates. It can go to glycolysis, which we'll focus now. It can also go to glycogen synthesis, which we'll also talk about. Alternatively, it can go to the pentose phosphate pathway. That will be the focus of Module 10. Or it can be used by other pathways. So let's focus on glucose 6-phosphate in the glycolytic pathway now. So the glycolytic pathway is summarized here, where D-glucose is metabolized to pyruvate. Remember, it's 2 molecules of pyruvate for every glucose molecule. Glucose is a 6-carbon molecule and pyruvate is a 3-carbon molecule. So again, the initial step is the phosphorylation of glucose. This phosphorylation commits glucose into metabolism with the cell. Glucose 6-phosphate cannot be transported back into the plasma. This reaction has a highly negative delta G naught. As we mentioned, glucose 6-phosphate is a branch point for metabolism, where this molecule can be committed to glycogen synthesis, the pentose pathway, or alternative metabolic pathways, or it can move forward in glycolysis. In the remainder of this preparatory phase, glucose-6-phosphate is isomerized via phosphoglucoisomerase to glucose-6-phosphate. You can see this double bond structure is specific to the isomerized form. Following that, fructose-6-phosphate is phosphorylated by an enzyme phosphofructokinase. This is our second ATP used so far in glycolysis. Fructose 1,6-bisphosphate is cleaved by an aldolase to generate dihydroxyacetone and glycero-3-phosphate. There's an isomerase, triose phosphate isomerase, that can convert these two products interchangeably. This next step, involving glyceraldehyde 3-phosphate, involves a dehydrogenation reaction using glyceraldehyde 3-phosphate dehydrogenase, and this will generate our first NADH in glycolysis. The oxidation step here is dependent on a cysteine residue in the active site of the enzyme. This allows for the formation of a high-energy thioester bond during the course of the reaction. The high-energy intermediate held in the enzyme active site readily accepts this free phosphate, phosphorylating the product to 1,3-biglycerophosphate and generating the first high-energy phosphate bond in this substrate-level phosphorylation reaction. In the next reaction, the phosphate bond is transferred to ATP using phosphoglycerokinase to generate an ATP. The 3-phosphoglycerate produced from this reaction is isomerized using phosphoglycerol mutase, which moves this phosphate group to carbon 2 of the molecule. This also allows for the removal of water to generate phosphoenolpyruvate and another high-energy intermediate. So once again, this phosphate can be transferred onto ADP to generate ATP in a substrate-level phosphorylation reaction using the enzyme pyruvate kinase. And this will generate pyruvate. Remember, after the cleavage of fructose 1,6-bisphosphate, this portion of the metabolic pathway happens 2 times for every 1 glucose molecule. So to summarize, for every 1 glucose molecule, we're able to generate 2 molecules of pyruvate, 2 molecules of NADH, and 2 molecules of ATP. Remember, 2 molecules of ATP were used in the initial part of the reaction. So the net gain of glycolysis is 2 NADH and 2 ATP. The pathway occurs with an overall negative delta G of approximately minus 22 kilocalories per mole. Therefore, this pathway does not go in reverse without the input of energy. So once pyruvate is generated, there are two primary fates of pyruvate. In the presence of oxygen, you have aerobic glycolysis, where pyruvate will be transported into the mitochondrion, accepted by the TCA cycle, and oxidized via oxidative phosphorylation on the electron transport chain. This piece we've talked about. Remember, the NADH generated by glycolysis must be shuttled across the mitochondrial membrane by either the glycerol 3-phosphate shuttle or the malate aspartate shuttle. Anaerobic glycolysis, or in the absence of oxygen, pyruvate is reduced to lactate. And the cell does this as a means of regenerating NAD+, so that glycolysis can still continue. And under these conditions, glycolysis only produces 2 ATP. Again, the 2 NADH that are generated by the initial glycolytic reaction are reoxidized to NAD+ by the reduction of pyruvate to lactate, and so the net result is only 2 ATP. This is really inefficient for the cell, and it also generates or can generate an accumulation of lactic acid. We'll look at this in a little more detail when we look at skeletal muscle metabolism. So here you see this reaction of anaerobic glycolysis where the production of pyruvate results in the generation of lactate, where pyruvate is reduced to lactate and NADH generated from a glycolysis can be oxidized to NAD+. This reaction by lactate dehydrogenase is reversible. Let's stop here and ask a question. Which of the following pairs of phosphorylated molecules contain an intermediate of glycolysis with a high phosphate transfer potential and one with a low phosphate transfer potential? So what about molecules with high phosphate transfer potential? That would be a phosphoenol pyruvate and our 1,3-biglycerol phosphate. So from this we can eliminate D. Both of these, the biphosphoroglycerate and the phosphoenol pyruvate, are both high energy molecules. We can eliminate E as pyruvate is not phosphorylated. That leaves us with B, which is the correct answer. Phosphoenol pyruvate has a high phosphate transfer potential, and glyceraldehyde 3-phosphate has low phosphate potential. Remember, glyceraldehyde 3-phosphate is converted into the 1,3-biglycerol phosphate to generate another high energy phosphate potential.